Hello everyone, my name is Arwa Raies. I am a senior data scientist at the Center for Genomic Research at AstraZeneca, and I am pleased to present our work on druggability prediction using machine learning. Druggability is an important concept that influences target selection and has a huge impact on the success of drug development. Druggability is defined as the probability that a target is amenable to interacting with a small molecule or monoclonal antibody, providing a desired therapeutic effect. It is important to estimate how druggable a gene is at the early stages of the drug discovery pipeline. However, predicting druggability using standard machine learning approaches is challenging due to the small number of known targets, lack of robust negative data, and extreme data imbalance. To address these challenges, we developed DrugnomeAI for estimating a druggability likelihood for every protein coding gene in the human exome. DrugnomeAI portal is an interactive and freely accessible online application integrating machine learning-based prediction around druggability likelihood across the human exome. The tool allows for viewing the druggability profile of a given gene across different models, drug modalities, and therapy area, as well as illustrating top features of druggable targets. It also integrates data from public sources around druggability. We developed 7 disease-agnostic and 8 domain-specific models to highlight genes for therapy areas and drug modalities. Our drug modality-specific models predict which genes have properties that make them amenable to modulation by small molecules, monoclonal antibodies, and PROTACs, as well as predicting druggability for oncology and non-oncology therapy areas. The model achieved median accuracy of 97% in predicting known targets. We tested combinations of gene labels, features, and classifiers, and the best performance is achieved using a gradient boosting model trained on all the features and using the tclin dataset, which consists of about 600 positive labeled genes. Our analysis shows that the majority of the top 5% DrugnomeAI ranked genes have been selected for clinical development, and half of those reached stage 4 of clinical trials. This demonstrates the model ability in prioritizing targets that have clinical relevance. As for the genes in the top 5% that do not have clinical evidence, we determined that these genes are associated with diseases using non-clinical evidence from the Open Targets platform. Additionally, we observe high enrichment based on exact Fisher's test between the top DrugnomeAI genes and high-ranked genes from large-scale PheWAS case control studies for binary and quantitative traits, illustrating the model's ability in identifying targets with genetic relevance. We observe high overlap between top DrugnomeAI predictions and validation datasets obtained from the Open Targets tractability data for small molecules and antibodies-based models, outperforming previously published tools. Feature analysis using Baruta algorithm shows features derived from protein-protein interaction networks from DG-IDB, String, InWeb, and Reactome are among the top contributors for druggability prediction, thus confirming the benefit of tackling the druggability problem at the system biology level. Other notable top features include monoclonal count, protein sequence length, RNA specificity from Human Protein Atlas, as well as processes from CTD, the Comparative Toxigenomics database, such as apoptosis, which is highly ranked for the oncology-specific model. As for the model development, DrugnomeAI architecture is based on MantisML, a framework that we developed previously that implements stochastic semi-supervised learning on positive and labeled data. To train the models, we compiled sets of known targets from various sources, including Ferris and triage labels. We integrated multiple data sources into the framework, resulting in more than 300 general and druggability-specific features, in addition to protein-protein interaction networks and pathway data. In conclusion, DrugnomeAI supports predicting druggability of novel genetic candidates at the earliest stages of the drug discovery pipeline. The work aimed to expand our understanding of druggability by illuminating gene properties that predispose their binding affinity and increase efficiency of target identification by prioritizing targets that have the most similar profiles of successful drug targets. I would like to thank my team at the Center for Genomics Research at AstraZeneca. The presenter and contributors of this work are employees of AstraZeneca. Thank you for listening, and if you have any questions, feel free to contact me.